Okay, so we are streaming on YouTube. If I could get a tech check on that just to make sure that it is streaming. Says it's done. It looks good to go on my side. Yep, we're good. But now I have to not listen to myself in two screens. Okay, so we are live on YouTube. Technical note for those watching on YouTube, we will not be taking questions from the YouTube audience. However, if you want to join us in the Zoom room while we are live on January 18th at 6:31 p.m. Eastern Time, if you want to join during this next 2 hours or so, there'll be a link where you can come join us in the Zoom room and ask questions within the Zoom room. And at the conclusion of the webinar, a few minutes before we actually end the full webinar, we will stop recording for YouTube so that anybody who is in the private room in the Zoom webinar and wishes to ask a question that they do not wish to have kept forever on the internet, there will be a brief moment at the end of the meeting for you to ask such questions. So now that we've got all the technical stuff, and it looks like we've got about 50 or so people joining us here on the webinar, let me welcome you officially to the HCMA's. I don't even know what number we're on. We should probably go back and count, guys. At least the 30th Bighearted Warrior Tour online, I believe. We started in 2020, and we've been doing these just about monthly ever since, so do the math. And we have a couple extra ones that we've snuck in there as well. So if this is your first time joining us, let me give you some housekeeping. We will be taking questions and answers throughout the evening by using the Q&A box found at the bottom of your Zoom webinar screen. We will not be taking verbal questions. Please do not raise your hand. We won't be looking at that. If you have a technical question about the functionality of the room or technical issues related to communicating with HCMA, our staff members are here to help you in chat. If you have any questions for our speakers this evening, you would use the Q&A box. So without further ado, I think I've handled all of that, and I launched the questionnaire, the poll. So if you can take a few minutes as you come in the room and do the poll at the conclusion of my talk, I will bring up that poll and we'll know who we're going to be talking to this evening. We get a diverse group of individuals here. Sometimes we get patients, families, industry, other clinicians. So when they do the survey, we'll know who we're talking to tonight. So with that said, I want to thank Stacey Titus Brown and Julie Russo from the HCMA team for helping with the technical assistance and administrative support of the Bighearted Warrior Tour. So they're going to be in the background. You're not going to see them on camera all night. They're going to go hide in the background and make sure that everything's running smoothly. And if you have any technical difficulties getting to this meeting, then you can call Julie. But if you're having technical issues, you won't be seeing that message anyway. So these meetings get a little long and they get a little technical sometimes for people. So just so you get the sense of the evening, we try to keep this kind of moving along in quick fashion. There'll be some Q&A. Try to keep it light and conversational as we're talking about very complicated topics. So with that, I welcome the team from Yale. And I think John Sendel is going to introduce the team to us, and I hand it off to John.
Thanks, Lisa. We're excited to do this webinar tonight. As Lisa said, I'm John Stendal. I'm a physician in the cardiomyopathy program here at Yale. I specialize in cardiac imaging and treatment of cardiomyopathies. So just going to introduce the rest of our team here. We have Dr. Nikhil Sikand. He's an advanced heart failure physician in our group. Also specializes in treatment of HCM. We've got Shaunak Sen. Dr. Sen is also an advanced heart failure physician, cardiologist, who also specializes in treatment of cardiomyopathies. And so the three of us see the bulk of the patients from a direct management. We also have an APRN who's not here today, who also works very closely with us. And then we have I think our group is somewhat unique in that we also have several specialists that we work very closely with. One of those is Dr. Rachel Lampert, who's a cardiac electrophysiologist, um, and has a special interest in um, uh, treatment of management of arrhythmias in the setting of cardiomyopathies. And she's an accomplished researcher in that field as well. So we'll hear more from her later. We've also got Dr. Aryamani, who's a cardiologist, but a cardiac geneticist as well. And so we work very closely with him, and he'll be talking later in the day. And he's a very accomplished researcher as well in the field of cardiac genetics. And he works very closely with Sarah Belsky, who's a genetic counselor. You see there as well. So that's our team. You'll be hearing more from us later, and so keep things short here. But I guess I'll turn it back over to Lisa for introductory comments, I guess.
So I'm going to give you the State of the Union 2024 abbreviated HCMA. Where are we now? What are we doing this year? Talk and make sure that everybody knows what they can expect on the HCMA this year and how they can get involved. So I'm going to show you a couple of slides. And while I am doing this, or if I bore you, you can go ahead and take the poll. Did I mention take the poll? Please take the poll so we know who's here. So we've got 75% of you, 25% need to jump on. Okay, so this is what we're going to be talking about tonight. If you signed up, you've probably already seen the agenda, but it'll take a minute to go over it. We're going to talk about myosin inhibitors and some current updated information on the MAVA trials. We're going to look at upcoming clinical trials. Also got some interesting news on that later. Exercise in HCM, which Rachel Lampert and I have been talking about for more years than I like to think about sometimes, but it's been a wild ride and things have changed a lot. So I'm looking forward to that. Genetics in HCM is critically important today, maybe more so than it ever has been, given the recent advances in gene therapies. And advanced heart failure therapies are near and dear to my heart, pun intended. And I'm sure we'll all gain a lot of knowledge tonight from all of these topics. I want to take a moment to thank the sponsors of the Bighearted Warrior Tour and the HCMA in general, Embria Pharmaceutical, Bristol Myers Squibb, Boston Scientific, Cytokinetics, Edgewise, Tenaya, and BioMarin. It's the partnership with pharma and industry that we have established over the years that allow us to bring you great programs, have great staff, and provide excellent advocacy services for the community. So we thank our sponsors, and we really look forward to continuing these great relationships. So this is all we have to do this year with a bunch of other stuff thrown in. So just to give you a high view, like what are we focusing on this year? What projects are the HCMA trying to get to? There's a couple of key dates. Number one, we are doing our first ever and hopefully to become an annual event, visit to Capitol Hill. We are going to talk to Congress and the Senate about issues that are important to the HCM community, which we're starting off with the topic of help us get diagnosed. So I'll talk a little bit later about the Healthy Cardiac Monitoring Act as a state-based initiative, but as a federal initiative, we are seeking, in addition to the Medicare exam, that it too include family heart health history questions so that clinicians can identify families who are at risk in a more timely manner. So that is what we are going to be focusing on Hill Day. And if you live in the Washington, D.C. area and would like to come join us on the 14th or 15th, there will be opportunities to do that very, very soon, maybe even tomorrow, where you can sign up and join us. There will also be, very shortly again, drop the links to HCM Awareness Day, which is the last Wednesday of every February. And we are going to be holding a webinar that night to bring the community together, to hear from our Center of Excellence partners, our industry partners, and really elevate HCM as only we can on a special day of the year to really look back, what have we accomplished in the past year. We are making remarkable progress in so many ways. So you can come and get caught up on that on February 28th and hear some amazing and inspiring stories. We'll be participating at the ACC conference in April. We're going to have an in-person meeting in Ohio, May 4th. We may be at the Heart Rhythm Society meeting. I'm not 100% sure yet. We may just be attending and not exhibiting. We will be at the European Society of Cardiology in England and the Heart Failure Society of America, which runs in concert with the HCM Society Educational Symposium. And then in October, we will be at the International HCM Summit in Boston. And for the first time ever, we're collaborating a patient meeting with the International Summit. So HCMA will be programming HCM night at the summit on Saturday night, and then our patient meeting will be Sunday, the 27th. So hopefully you'll see an agenda in the next month or so on that. And you can start registering and making your plans to join us in Boston this October. And then, of course, we end the year with AHA, and this year it's in Chicago. In November, I'll be cold. Okay. So, talked about HCM Awareness Day. I'm going to kind of try to go fast here, but during the day, we're going to do 5 legislative briefings, kind of lunchtime briefings on our legislative initiatives. We've identified 4 of the states that we're definitely interested in. So, I'm up for passionate volunteers to come up with state number 5 if you have one. We can talk about maybe that being a one added to the list. So, we're going to do the briefings during the day, take a little break, and come back with some nighttime content. So, what are the legislative opportunities that you have right now and you can potentially even do from your home? Um, Last year, we had a whole bunch of HCM Day resolutions and proclamations run through different state houses. Connecticut was one of them. And I just say that because Yale's in Connecticut. So that was great. But sometimes these are only 1-year proclamations or resolutions. In Ohio, we are actually a state law to be recognized. So if you want to get HCM Awareness Day to be recognized by your state, you can call the office and speak to Julie, who you saw just a few minutes ago, and she can tell you exactly what you need to do to get your state to consider a proclamation. Typically, they only like to get these things about 30 days in advance. So this is the time to do it. So we can help you get that set up. This was my visit to the Capitol or to the State House in Michigan, in Lansing, Michigan. I literally brought the heart of a Michigan woman who transplanted from HCM and literally put her heart in the hands of the lawmakers. And we should be getting a bill number very soon for the Healthy Cardiac Monitoring Act from Representative O'Neill's office. And no way to get somebody on your team better than literally putting a heart in his hand. So we don't always have to be that dramatic, but sometimes it gets the point across. So, what is the Healthy Cardiac Monitoring Act? It's super simple. It's asking the right questions in the well child exam to help identify families who are at risk for hereditary heart disease, not only HCM, but other genetic heart disorders. So it's about asking questions, it's about educating physicians, it's about educating the public about why we're talking about family heart health history. We've got to start somewhere with a structured conversation and a structured path forward for better identification. And we've spent 3 years developing this sample policy, and in fact have got the endorsement from the American Heart Association to go ahead with it. They're not taking the lead on it, but they are welcoming us to take the lead and they'll join us in this endeavor. So if you want to get involved, please let us know. If you have contacted the HCMA, I'm pivoting now, anytime in the past 27 years, I probably took notes on our conversation. Later this month, maybe next month, we will be moving to submit for IRB approval of our patient registry. So all of that data that we've been collecting, we're going to be able to look at in a new way. And as you come on to the HCMA, you'll be consented into the patient registry. We'll be reaching back to you to consent you into the registry. And then we'll have an opportunity to look for those gaps in care within the HCM community and really work on creating concrete projects that we can move forward in a positive, productive way to bridge all the gaps that exist right now, whether they be in clinical care, emotional support, financial support. We're trying to figure out a lot of different things that we could do to help the community. Um, if you are a member of the community and you are having financial difficulties and you need support in getting to a center of excellence or a transplant program, we do now offer the Lori Fund where we can offer scholarships of up to $600 per person per year to travel to a center, trains, planes, automobiles, food on the road, hotel rooms, whatever you need to get to the care you need. We are trying to remove burden wherever we can. And this one we can do if you financially qualify. We've been able to give out a whole bunch of scholarships. And in our annual report, you're going to read a lot about those who benefited from this fund that was set up in my sister's memory just 2 years ago. Tales from the Heart, a podcast, doing some new versions of the podcast. If you're a patient, if you're a clinician, if you're a researcher, industry member, and you want to share your story with the HCM community, I'm doing 2 new podcasts, 30-minute formats, recorded on Friday, dropped during the week, so they're not live, so you can make a mistake and we can edit it out. Don't fear. But we want to share more stories and more perspectives. So if you want to share your story with the community, think of it as the global community. Let us know. Talk to Julie or Stacy and get on the list. We'll set you up. We want to have these conversations. So we went through this already. This is kind of a redundant one. We are going to be doing at least two other in-person meetings this year, but we haven't announced where yet. Discussion groups. More discussion groups are being added. We're adding another transplant group. We will have a pre-transplant discussion group and an all transplant discussion group once a month. So you're going to have an opportunity, if you're in the pre-transplant process, to have contact with your peers. I think it's going to be the first Monday of the month, and then the third Monday of the month is going to be the total group. So if you want to see the post-transplant people, you will be able to join us in that second group. But we know that pre-transplant group, you get a little lonely out there. We want to provide as much support as possible as you wait. So we've added the second group. So we're going on the map. We're going to go see a lot of people soon. So we're really looking forward to getting back in person. Like I said, Ohio's definitely happening. Utah's definitely happening. I just don't know the date. So we're going to get out of the East Coast and start heading west again. Join us in Boston. We talked about that at the start. And I thank everybody for their time and attention, and our wonderful board, contractors, staff, volunteers, and partners. It takes a village to make all of this happen, and I thank you for all being part of my village. And of course, thank you to Brandy, my donor, without whom I wouldn't be here today. So let's bring everybody back up on camera and let's see. I'm going to end the poll. It's going to be representative. We've got about 74% telling us who they are. So let's take a look. 39% are Northeast. We've got some Southeast, Southwest, Midwest, West. So we've got the country handled, and we're sticking. Oh, we have another. So whoever the other is, you have to write in chat what other is that I did not explain here. So other person, please put it in chat. I'm always curious. It's happened before. So what is your relationship to HCM? So 63% are patients, 13% are a patient and a family member, and 11% have a family member. 5% are medical providers, 5% are industry, and somebody's just curious. Other rule, curious. Why are you curious? Give us a hint in the chat box. I'm always fascinated. Okay. Have you or your family member had any of the following? Diagnosed within the last 2 years, 21%. So this is the learning phase. We understand that that's a really challenging time. 58% are on meds, 34% have an ICD, 18% have had septal reduction, 29% have had AFib, 16% have lost a family member, 55% have had genetic testing for HCM. That should make our gene folks happy here tonight. And there are people considering therapy. 5% have been advised to get an ICD, but haven't done so yet. And 11% have been advised to consider septal reduction therapy. So there's some decision-making going on, in fact, gathering tonight. Are you considering a new medication or device at this time? 29% of those watching are considering changing their management, and 71% are not. And 58% have been to a center of excellence, 34% have not, 3% are unsure. That one's easier to be unsure about. And if applicable, are you a patient of this center? So you have 5% of your patients are here tonight in the population. And you've got a bunch of people not currently being seen, so they're shopping for doctors. No pressure whatsoever. Okay. So we have a diverse group here tonight, some new people, some people who've been around HCM a little bit longer, and I'm sure they're going to have some interesting questions. So remember, you can drop your questions for our panelists in the Q&A as they're speaking. If your question gets answered, you can withdraw the question, but we ask you kind of keep up with it. We're going to do a brief break after each talk for a little comment, and then all the rest of the questions will be held to the end if they're not on topic. So with that said, let's go back to the agenda, and we are going to kick off with who's kicking off? Nikhil, I'm going to kill your last name, Sikand, right?
Yeah, that's right.
Sorry. It's been a long day and
No
I'm
worries.
tired.
I will share my screen.
Perfect.
Okay, great. So thanks, Lisa, for and everyone at the HCMA for hosting us here. It's really a pleasure to be with all of you tonight to talk about what I think is a really exciting topic. My name is Nikhil Sekand. I'm one of the cardiologists at Yale, and I specialize in inherited cardiomyopathies. And I'm going to tell you just a little bit about what you need to know about cardiac myosin inhibitors. So before we get to the specifics of cardiac myosin inhibitors, I'll just talk about briefly about the history of major developments in the field of HCM, because I find this really fascinating, and it dates back several hundred years to some of the first published descriptions of HCM in the 1800s and 1869 in a journal in France called the Paris Gazette by a French physician named Henri Louisville. But it wasn't until the 1960s that therapy was really described in the medical literature. In 1961, the first surgical resection or myectomy was performed at the NIH. And several years later, the great physician Eugene Brunwald described, also at the NIH at the time, described use of beta blockers for patients with hypertrophic cardiomyopathy, now a standard of care that I'm sure many people are here are familiar with. In the 1990s, septal reduction with alcohol septal ablation was developed and first used in the United Kingdom. And in the early 2000s, the medication disopyramide, which others may be familiar with here, was shown in some studies to be effective as medical treatment. But really, I think some of the most exciting medical developments have come in the past decade, and specifically in the past couple of years. And in 2022, the cardiac myosin inhibitor mavacamten, commercially known as Camzyos, was approved by the FDA as the first ever medication specifically studied in patients with hypertrophic cardiomyopathy and targeted to the mechanism behind their disease. So I think it's worth covering, not to be too technical here, and I hope I'm not. I'm happy to answer questions afterwards, but how do these drugs actually work? Well, they work on a cross-link of proteins called actin and myosin. So some people may remember back to high school biology, or others may have science backgrounds here, but actin and myosin are proteins in the cardiac sarcomere, and that's really the functional unit of heart muscle. So if you break down heart muscle, this is kind of the unit that drives it. And they connect and they form these bridges in the cardiac muscle that provides contraction. You can see here on this graphic, this is exactly where this happens. And in hypertrophic cardiomyopathy, there's excessive formation of these bridges between the actin here and the myosin. And that causes thickening, impaired relaxation, and leads to some of the symptoms that patients have. And what the cardiac myosin inhibitors are designed to do is they bind to this myosin and inhibit the excessive connections that theoretically will allow for more relaxation in the relaxed position you can see here on the right. So what is the data for using this medications? Because I think whenever you're considering a new medication, especially one that's recently FDA approved, you should always or your provider should always be familiar with what kind of data is out there. Well, a major trial that was published called the Explorer randomized control trial. So this was published in the Lancet. And I'll just give the take-home points here. But in that study, they studied patients, comparing them from mavacamten to a placebo, meaning a drug that did not have any medication in it. And in that study, twice as many patients achieved improvement in exercise capacity when compared to the placebo. And they had a reduction in their LVOT gradient measured on the echocardiogram on average 47 millimeters of mercury, which is a very significant amount. And 2/3 of the patients who were taking mavacamten in that trial had improvement of their symptoms in some way. So sort of the graphic here, 2 out of 3 patients gained some benefit when taking this drug compared to placebo. The Valor randomized control trial was a later published trial, and this looked at a slightly different population of patients who had more severe symptoms of hypertrophic cardiomyopathy, specifically obstructive hypertrophic cardiomyopathy, and likely were going to be candidates for a procedure called septal reduction therapy. And in this study, over a period of 16 weeks, 4 out of 5 patients who were on mavacamten experienced some improvement in symptoms, and a very significant amount were able to avoid a surgical procedure. So if you had mavacamten, 77% avoided the surgical procedure versus 18% while on the medication. So 4 out of 5 of these patients gained benefits. And finally, you may have seen some of the headlines over the holidays that a trial just announced for Sequoia, which is studying aficamten, which is a second-in-class medication of a cardiac myosin inhibitor, the results of which are not yet published and available for our peer review, at least from our center. But we do know that the results of that trial are positive. So the medication did demonstrate significant improvement in exercise capacity, again, for patients with obstructive hypertrophic cardiomyopathy. But perhaps as important as understanding the data is understanding what side effects you might have if you were to start on one of these medications. So in the trial, the most common side effects were dizziness and fainting. But importantly, because of the way that the drug is metabolized, there's significant potential for interactions with other medications that you may be taking. For premenopausal women, there is a potential for fetal harm with this medication. So women who are pregnant or planning on getting pregnant should not take it. And those who are of childbearing age should use contraception and be encouraged to have regular pregnancy tests, certainly a pregnancy test before beginning the medication. There are serious but rare possible side effects, and the most important one being a reduction of ejection fraction and development of heart failure. As such, the drug is monitored under something called the REMS, which you may come across if you're on or considering being on this medication. The REMS is mandated by the FDA, and it stands for the Risk Evaluation and Mitigation Strategy. And it's specific to each drug that it's being rolled out for. In this particular case, the REMS program involves regular monitoring with echocardiograms while you're on this medication. It involves clinical monitoring for adverse effects you may be having. It involves an examination of potential drug-drug interactions, and only certain pharmacies can distribute this medication. And ultimately, patients are enrolled in registry and are monitored long-term, as this is a first-in-class medication. So I think I'll cover just a little bit of how our program works for cardiac myosin inhibitors, and that might give you an insight into what it might be like to be initiated on one. So we see patients for an initial visit here, and during that visit, we typically discuss all treatment options, and that discussion is with an HCM cardiologist, typically Dr. Stendal, myself, or Dr. Sen. We obtain all initial imaging and review all imaging in person, including an echocardiogram or other testing. And then we participate in shared patient physician decision-making on whether Camzyos or mavacamten might be a medication to consider for your particular form of hypertrophic cardiomyopathy. If the patient and us agree that this is a good option, we'll move to the initiation phase. And typically at that point, you'll meet with our other team members, including our APRN and pharmacists, and we'll enroll you in the REMS program, go through insurance authorization and prior authorizations, as well as understand payment obligations for patients. And ultimately, the medication in our case can be dispensed through a specialty pharmacy at Yale. And then following that is the monitoring phase. And what we encourage and do here is high quality echocardiograms, meaning done by our trained echocardiographers who are trained to look at the hearts of people with HCM. And these happen at week 4, 8, and 12, and then every 3 months after that. So there are fairly frequent echocardiograms as part of this medication. And finally, check-in visits are done for symptom assessment or dose titration. We often do these visits virtually for our patients, unless they need to be seen in person, of course. And then ongoing assessments to decide whether the medication should be extended for a period of time, whether patients should move on to alternative therapy, or any other concerns patients have. I'll just end by talking about the future, and hopefully this will segue a little bit into Dr. Stendhal's talk, but we are also enrolling in clinical trials at Yale, specifically for cardiac myosin inhibitors, Maple HCM and Acacia HCM. Maple HCM is for patients with hypertrophic obstructive cardiomyopathy, and it's studying the drug aficamten and comparing it to metoprolol, which is considered the initial standard of care. This study is sponsored by Cytokinetics. And we're also part of the Acacia HCM study, and this study is looking at the medication aficamten, a cardiac myosin inhibitor, in patients with non-obstructive HCM, which is a form of the disease which up until this point has had very limited therapeutic options. So those are two trials that we're doing right now. So to summarize, cardiac myosin inhibitors are a new medication. They're specifically designed to target proteins in the heart that are involved with the development of HCM. Mavacamten, or Camzyos as it's known, is the currently only FDA-approved medication. However, the medication aficamten is in clinical trials, including a recently positive clinical trial. The medications have been proven to improve quality of life and symptoms in selected patients with this disease. The drug is taken by mouth daily, and it definitely involves close monitoring by your entire clinical team, and preferably at an HCM center of excellence. And finally, cardiac myosin inhibition as a medication class may have broader applications in hypertrophic cardiomyopathy and also in other forms of cardiomyopathies, as well as heart failure, and is being studied for these indications. Thank you for having me. This is our contact information, some information about hypertrophic cardiomyopathy that we offer, our Twitter handle, and our contact information.
Is it Twitter or is it X?
I'm sorry, X handle. I should have known.
I know I can't get used to that quite either. Okay, so we do have 2 questions in the hopper. If you have any other questions, type now because we're going to go quickly through this. I'm going to read you the question and then I'm going to reword the question and then I'm going to ask for a response. The question is written is, when might Camzyos exit the REMS program, thereby reducing the close tracking? So I'm going to rephrase that to say, this is an FDA requirement. There isn't an exit door for them to choose, and patient safety is paramount. Even though the consequences of dropped ejection fraction are rare, they are critically important to find in a timely fashion. People have died. This is not something to be taken lightly, and this is just not an inconvenience to make everybody miserable. There are safety signals here. So having said that, what do you think?
Yeah, I mean, I completely agree. I mean, the FDA, when placing a REMS on a medication, it's a big decision, and certainly one that's taken very seriously. As I mentioned, and you just mentioned, the cardiac myosin inhibitors do have the potential to lower your ejection fraction and in rare cases cause heart failure, which could be fatal. So because of that, these echocardiograms are monitored on a regular basis. And the FDA then wants to know, long term, exactly what's happening in the real world with these patients, because the patients in the clinical study are obviously not the same as the patients in the real world. So it remains to be seen, but I would say that probably this form of monitoring will be around for the foreseeable future. So patients who are considering this medication should sort of plan to be monitored in this way.
Okay. So the next question is, and I don't know the answer to this because I've never seen anything published on it. Does this medication interact with testosterone?
Ooh, that's a good question, and I don't know that. If any of my colleagues know, perhaps Dr. Stein or Stendahl, if you're aware of any of those interactions, I don't know that question. I'm happy to look into it and get back to whoever answered it, and we can get a better answer about that. But I haven't had experience with
that. Anybody got data on that? I've not seen anything published, discussed. I don't know that it has any traceable impact. I can go back to the manufacturer and sponsor and check with them. So
you can say it's not on the list of contraindicated medications.
It is definitely not on the list. Yeah. Typically, when we have those questions, we bring them up specifically with our pharmacy team, and we look into every single one of those questions. And if we need to go back to the spot to person wireless split and speak with them about, what they know.
That's the mechanism of action you wouldn't expect to be
Okay,
in.
I think we have somebody interesting posting questions as an anonymous attendee because they said no, MAVA doesn't interact with testosterone. So I'm wondering who that's from. I don't know who it's from. If you are from BMS and you have my cell phone number, let me know who's answering that. But I don't know that that's true or not, but I will confirm that. And T Webb, who asked the question, if you call the office tomorrow and leave your name and number, we'll make sure we get back in touch with you. Does it interact with estrogen? We're on a hormone kick, people. Does mavacamten interfere with estrogen? I'm unaware of that as well. I promise I will do a dive into hormones and Camzyos with the scientific group behind the scenes at BMS and get you any information I can. So question is, does AFI also require echoes? Well, aficamten is not approved by the FDA at this time. It is a drug that is in clinical trials, and the utilization of the drug is dependent upon the protocol, which includes a great deal of surveillance because it is only a trial. Any other comment on that?
No, exactly. So any patients that have received aficamten to this point have done so in the trial. So it will be, the sponsor clearly is going to present this data to the FDA, and again, they will make the decision on whether or not the drug warrants a REMS.
Great, thank you. Have AFI tests shown any heart failure risk? So the top line readout data came out 3 weeks ago. It is public. It is minimal information, and it looked very promising. And there were not any cases of drop in ejection fraction under 50% that were reported in the top line. Does anybody have any deeper knowledge they wish to share?
No, I think that's what everyone knows publicly at this point, and we're going with that. But I think before advising patients, obviously we'll have to review all the full trial data ourselves and understand sort of what the deeper numbers show.
And the last question, I can take this one. What is the schedule for pediatric trial? All I can tell you is it is in deep discussion and it should be coming to you later this year. No more specifics than that, but there are a lot of people very, very interested in this particular question. And I assure you, we are hoping to see this come to life very soon. With that, we have cleared out the myosin inhibitor questions for now. I'm sure there will be more at the end of the evening. And we are going to pivot to our next speaker. John, you're up. Take it away, Dr. Stendel.
Hi, I'm just going to share my screen here. So
Almost there.
I think we got it.
One more step.
Okay, can everybody see this now?
You're not in present mode yet.
Oh, let me go.
And now you want to change your display settings so that we go to the top display setting and swap because you've got two screens.
Yes, let's do that. Yeah, that's it.
Swap. Here you go.
There we go. Okay.
You're off.
Sorry about that. Okay, so this, I'm going to talk about research efforts. A lot of this will be kind of dovetails into what Dr. Sekand was presenting. Talk about our ongoing clinical trials, but also talk about some other efforts here. So, as I mentioned earlier, we have a group that involves not only cardiomyopathy specialists, we also have imaging specialists, specialists in cardiac genetics, and also electrophysiology. And so, bring that group. Our group meets together regularly, and we have numerous forums or research forums to talk about patients, talk about genetics, and that's part of our work in the cardiomyopathy program here. Some of the ongoing formal research projects, we have several efforts in clinical and translational research. One is the SHARE registry. That's an ongoing effort. this is a research community that's dedicated to advancing the understanding of inherited cardiomyopathies. And the focus is hypertrophic cardiomyopathy, but it also involves work with dilated cardiomyopathies and also arrhythmogenic cardiomyopathies. It's a clinical database of patients. So we have sites in multiple sites in the USA, Europe, Australia, and South America. And the participants were one of the sites, but the participants at those sites basically pull patient data and have this database for study. And it's been very helpful, and this has been a very fruitful endeavor. And there's a lot. If you go to their website, you can see there's been lots of publications that have come out of these efforts. So we have ongoing efforts to analyze the data, and this is a continuous effort. Participants in the share registry from the different sites submit proposals to do research projects, and then the entire group kind of analyzes the data that comes out of those efforts. We currently have an effort to collect blood samples from our patients with HCM, and those are being, there's a specific NIH grant that's being conducted to analyze the genetics in those samples and connect that with clinical outcomes, clinical characteristics and outcomes. So that's a large effort that's going on, ongoing effort in our group. We've been a member of the share registry for many years now. So additional projects. One is Live HCM. Rachel Lampert is heading up our effort here at the project, so she'll talk about that in detail. So I won't talk about that as much. We have some ongoing efforts with the Stuart Campbell's laboratory, the Integrated Cardiac Biomechanics Laboratory. Stuart is a bioengineer with an interest in cardiomyopathies and inherited cardiomyopathies. So he has techniques to study the biomechanical properties of heart tissue. That's quite interesting. So, and he also combines that with genetic analyses and cardiac tissue engineering. So he's able to, from blood samples, we've done this on a few of our patients who consented for study, but he's been able to culture actually beating heart tissue from blood samples from the patients, and then use that tissue to develop to measure the contractility of it and the different properties of the tissue that they create in a petri dish, and use that to better understand the mechanisms of disease in HCM. So he's done several studies on families that we see in clinic. So that's ongoing work. I'll talk briefly about finish up with the clinical trials here. And this is basically ties into what Dr. Sekand was talking about earlier. So we have numerous trials with mavacamten and aficamten that are currently in progress here. Valor HCM was one of the current projects with current trials with mavacamten, and that's basically to evaluate the effect of mavacamten on decreasing the use of septal reduction therapies, such as surgical myectomy or alcohol septal ablation. So it's a phase 3 trial. It's active. This trial is active, but it's no longer recruiting. So we're in the data collection phase of this trial, and it should be wrapping up by next summer. Initial results, though, were positive, and that mavacamten reduced the proportion of patients proceeding to septal reduction therapy. There's numerous other data were associated with this that were generally positive. So, as Dr. Sekand mentioned, so this is one trial that's going on, but not recruiting. MAVA LTE is a long-term extension of the Explorer trial that was mentioned earlier, and this is just ongoing monitoring. I think so. The results from this is that the benefits of mavacamten are sustained at long-term follow-up, and the safety as well. So the initial signal. So that's promising. The other trials that are going on are with aficamten, and so one of them, as Dr. Seekan mentioned, is Maple HCM, and this is basically to compare aficamten to the standard of care of metoprolol, and so really comparing those two medicines. And so this trial is ongoing. We're recruiting for that, and so the contact for that for patients in our area who are interested, there's the coordinator for that is Robert Cantafio. His contact information is listed there. We can provide that at the end as well. Another trial, which was also mentioned earlier, is the Acacia HCM trial. And this is in non-obstructive HCM. And so these are patients that don't have an outflow tract obstruction, but have basically diastolic dysfunction related to their hypertrophic cardiomyopathy. And this is a really common problem. And, I think one of the important findings of the initial work is that the myosin inhibitors have been very, very effective at not only reducing the obstruction, but also treating the diastolic dysfunction that happens in hypertrophic cardiomyopathy. And so, so we're really excited to see what what these trials end up showing. But we're very optimistic that these would be have some very, very pertinent findings. And so, again, this is with non-obstructive HCM. This trial is also recruiting right now. Those in the area that are interested can talk to Robin Smith. Her contact information is listed there. So we're currently enrolling for that trial. Forest HCM is another trial that's ongoing. This is came out of the Sequoia trial that was earlier. Dr. Seekan just mentioned that the kind of initial data are coming out of that trial now. This is the long-term extension, basically to see that study the long-term safety and tolerability of aficamten. So this trial is really only for people who have completed the Sequoia trial or other trials. So this is not one that we're enrolling in specifically by only by invitation. So with that's about all I have. I guess we can answer any questions. I assume a lot of these will be similar to what the last round was, but happy to answer any questions at this point. I can put up the contact.
My mute wouldn't come off.
No. Do you want me to stop sharing or?
Yeah, we can stop the share now, and we have some questions posed for you. Interesting question. Are you doing any research or testing for non-invasive mitral valve repair and replacement?
I guess I'm maybe the heart failure doctors will be. I know that there's ongoing work studying that, specifically in HCM. I'm not aware of any efforts that we're conducting here at Yale, but that's more of an interventional approach, and sometimes non-invasive surgeries, or minimally invasive surgeries. I don't know. Heart failure guys, do you have anything to add?
No, I agree, John. I don't think anything specific in the HCM population in our center at the moment. I know there's a lot of exciting work looking at minimally invasive or percutaneous options for mitral valve regurgitation. I think in the context of HCM, it's important for all of us to recognize that we have to kind of decouple obstruction from mitral regurgitation, and that's really important as a first step. And I think that's an important first discussion. If there's obstruction, is there any way to relieve that? And what is the mitral regurgitation secondary, or is it a primary process? I think those are important things to talk about with our patients.
And in many cases, so if you treat the obstruction properly, you can solve the issue in mitral valve.
So the answer is we're unaware of anything. I know a couple people were using mitral clips, but other than mitral clips, I don't really know anything late breaking in mitral valves with HCM, and the mitral clips were only used on the elderly.
I was gonna say, we certainly have an active mitral clip program. We're very active, transcutaneous structural valve program in general, very active with the mitral clip, but to what extent it's been used in the HCM patients, I'm not sure.
So we have a complicated question, and I am going to say that we have to remember when we're dealing with trial drugs or labeled drugs. The rule book is a little different, and what we know is a little bit different. So questions about trial drugs may be premature because we're still learning. That's why it's trial. So the question is, I'm going to take off the first part of it for the narrative. Is there any information on aficamten side effects and drug-drug interaction? And is there any way to compare that at current time with the same list for mavacamten? Anybody want to try that one?
Yeah, I think the with aficamten, some of the the thought is that I think the initial thoughts were that it would have fewer drug interactions than mavacamten. But I think we're still finding out from the trials, exactly how that's going to play out. I think, the trial. I think the data we have right now is rather limited. I know, Dr. Secan, do you have anything to add to that, specifically for aficamten?
No, I mean, I think it does have a slightly different mechanism of action than mavacamten, and probably less liver metabolism than mavacamten. And many drugs are metabolized through the liver that patients may be on. Again, however, given that we don't have the full clinical data, this is really just theoretical at this point.
One of the main differences between aficamten and mavacamten, I said there's slightly different mechanisms, but the half-life of aficamten is much shorter. And so, with mavacamten, we had a schedule where it was make an adjustment, and then it was weeks later. And I think that potentially is, because the medicine would take a while to build up, and it also sticks around longer with mavacamten. With aficamten, it's cleared from the system more quickly, and so there's a faster buildup. And so so those are issues, or potential changes, and that generally the shorter half-life gives more flexibility.
So there's another question. I'm not particularly thrilled with the wording because it's a little confusing. So I'm going to reword your question. They want to know if there's positive side effects from myosin inhibitors. And I'm going to start you off easy because that's a softball. Like, yes, we have reduction of gradient. We have improved muscle compliance when you have extended use in some. Some patients have normalized EKGs after longer periods of dosing. But reducing atrial fibrillation, removing arrhythmia risks, ventricular arrhythmia risks, what do we know? What does it do? What doesn't it do?
But I think, Lisa, feeling better is, the most important impact that
it's a pretty big thing.
That the MCIs have had.
Is there any other positive result of a myosin inhibitor that we're seeing signals of or have proof of?
I think that, for me, I think the treatment of, it seems like in the patients that are symptomatic, it's usually either an outflow tract obstruction, severe diastolic dysfunction, or both. And these drugs seem to treat both of those problems. And so I think that we'll find out for real with these trials in the non-instructed setting. But it appears that I think that that's probably almost equally as important, that it's treating the diastolic side as well. There's some initial signals that thoughts that these may affect remodeling and stuff, but I don't that that's more, I think, speculation at this point. Not
Minimal data, but we have seen some signs, and
minimal data, and the effects probably not substantial for the time period that they've studied.
we are all learning together in live time, which is why it's really important to follow everybody real closely. Okay, I'm going to move on to our next speaker. Rachel, let's talk about exercise and HCM again, because we know we love to have this conversation.
All right, great. All right, you're seeing just the main slide.
You are good to go.
Okay, thanks so much for the opportunity to talk today about exercise and hypertrophic cardiomyopathy. So their questions about exercise and HCM really come from two directions. For most people with HCM, like most Americans in general, it's really about getting more active. So is it good for me to exercise? Is it safe for me to exercise? How much exercise can or should I do? How can I increase the amount that I'm doing right now? Then we also have, from the other direction, people who are already exercising intensely, competitively, get diagnosed with HCM. And from that direction, we really want to talk about how safe is high intensity or competitive exercise and sports participation. So how much we know that patients with HCM are less active than the general population. So this was from a study that Dr. Day, when she was at the University of Michigan, did in conjunction with the HCMA. And they found that the HCM patients were less active at work and with recreation, although many of them were active and some were even doing vigorous competitive sports. And they compared this to population data from the NHANES study. They found that many of the patients, though, had purposefully reduced their exercise due to advice they had gotten from different directions, and this had a negative emotional impact in a lot of people. Similar findings came from a study out of Australia. They found that only 45% of HCM patients met the recommended guidelines of 150 minutes of moderate to vigorous activity per week as recommended by the American Heart Association, what we're all supposed to do. And 16% had been advised, again, from all different directions, not to exercise at all. So in order to start to address the safety of getting back into exercise for people who weren't exercising now, Dr. Day and her colleagues did a study called RESET, in which they took 136 patients and they randomized them either to usual activity, just doing whatever they were doing now, or to exercise training. And this was a structured home-based exercise program that they worked with each patient to individually prescribe them their exercise prescription. And this was a 4-month study. And what they found was that those in the exercise group were able to get into better condition. So peak VO2 is a measure that we can get from a cardiopulmonary stress test where we look at, it really measures how physically fit a person is. And they were able to increase their physical fitness. And in a questionnaire type of assessment, they found that they did better and felt better as well. And there were no arrhythmic events in the individuals who had been randomized to the moderate activity group. And based in this study, the most recent guidelines, which came out in 2020, now recommend that for most patients with hypertrophic cardiomyopathy, mild to moderate intensity recreational exercise is beneficial because it improves fitness, functioning, and quality of life, and improves overall health in keeping with physical activity general population. So how do you get started exercising? You know, I think the most important thing is just like anyone with heart disease, or really anyone at all, you don't go from, you know, a couch potato to the 5K in 0 to 60 seconds. So you really want to, you want to get there slowly. And most importantly, you want to talk to your doctor about how to get back into exercise. But there's really not a ceiling. There's no reason not to get back started exercising, but you just want to get there slowly and steadily. So I want to now switch gears and talk about the other end of the spectrum. For some people, the question when they're diagnosed with hypertrophic cardiomyopathy becomes, how much exercise can I do? How hard can I work out? What type of exercise? Is there an amount of exercise that's too much? Can I do recreational exercise? How much recreational exercise? Can I actually compete? Can I run? Can I swim? Can I play soccer or basketball, which are the things that I love to do? So prior to 2020, there were guidelines out there that said patients with hypertrophic cardiomyopathy should not be doing vigorous exercise and should just be doing what we call 1A, which is the lowest intensity, lowest dynamic, lowest static load, and basically includes things like bowling, golf, riflery, things that you might not even think of as sports. Our elected officials like to do this, but these types of sports, but most of us and our patients like to do more active things. So luckily, those guidelines, even as of 2020, have started to change. And I'm going to talk about some of the data that has led up to this. But most importantly, the 2020 guidelines for HCM now talk about, when they talk about athletes, they say for athletes with HCM, a comprehensive evaluation and shared discussion of potential risks of sports participation by an expert provider is recommended. So no longer are the guidelines sort of saying, you can do this, you can't do this, you're in for that, you're out for the other thing. It's really now a shared decision to talk about between a patient, an athlete, and their HCM expert physician about how much they should be doing. And based on some data that I'll talk about in a minute, they now describe participation in high-intensity recreational or competitive sports as something that may be considered, again, after comprehensive evaluation, shared discussion, and again, with an expert provider, that this is something that may be considered. So what are some of the data that has led into the change in thinking? This is a study that we did here at Yale that was a national, in fact, international study that asked the question, will individuals with HCM who are exercising vigorously have a higher rate of arrhythmia-related events than those not exercising vigorously? And this is really getting at the question of the concern that the original guideline writers had. The reason why HCM patients were restricted to these very low intensity sports was really concerned that these higher intensity sports might lead to arrhythmias. And so this was the question that we posed in Live HCM. And there may be people on the call that have participated in HCM, in Live HCM, are participating. And first of all, I want to say thank you if anyone is on the call that is in this study. So who was in the study? We enrolled people with, most of them were people who had HCM. A handful, it came out to 8%, were people who had the gene for HCM and a family member who had it. We enrolled people from age 8 to 60, and whether or not you had an ICD. We enrolled people at any level of exercise because the idea was this was a comparative study. So whether you like to read, to run, to walk, or sit and read a book, whatever you like to do, we enrolled you in the Live HCM study. And so if people were in it, they recognize these questionnaires. At baseline, we got all the clinical information about people like any study. And then we also asked the individuals participating to fill out a questionnaire relating to their activities. So first they just checked off the things they were doing and things that they were doing. We then asked them, so if you do like a water activity, how many hours a week, how many weeks per year, et cetera, so that we could really get a sense of what people were doing so that we could categorize them as either vigorous exercisers or non-vigorous exercisers. Then during the years of the study, which was 3 years for each person, although we actually have an ongoing long-term extension, but the basic study was 3 years, we asked you every 6 months to tell us how you were doing. Had you had a cardiac arrest? Had you passed out, been hospitalized? Do you have an ICD? Any new test results over the, and so we sent these questionnaires to people over the course of the study. So the first data from this study, the primary results came out in May of this year. So overall, and Lisa and I we have done some podcasts and what have you. Some of you may already be familiar with the results, or if you were in the study. So overall, we ended up with 1,660 people who both enrolled and followed up with us. These were about 2/3 male, 1/3 were female. We had 122 children under 18. We had 149 of young adults, and then the rest were young to middle-aged individuals. And 92% of them did have overt HCM, and the other 8% had the gene without the actual changes to the myocardium. So of these 1,660 people, we had about half of them were exercising vigorously. So of these, about half of them were exercising vigorously and competitively, and or somewhat more than half of this group were vigorous, but they were not competing. Then we had 709 people who were exercising moderately, and 259 people who were less active. And one thing I will say is this is a higher percentage of vigorous people than Americans in general, and I think this is probably because vigorous people wanted to be in the study, but we did have a nice range of people from the most active to the least active. So what we were looking for was to see whether the more vigorous group compared to the non-vigorous group was more likely to either have an episode, to either die or to have an episode of cardiac arrest, passing out, or ICD shock if you did have an ICD. And what we found was that basically the two groups were essentially identical. The non-vigorous individuals, 4.6% of those people reached that endpoint, and among the vigorous, it was 4.7%. As you can see, the lines are pretty much one on top of the other, and there was really no difference in the rate of these outcomes between these groups. We also had, we did look at the group of people who were younger, and really, we had 42 young people in high school and college who are participating in varsity sports. And I didn't put them on the slide, but when we looked at those people, they did not have a higher risk compared to the other people of their age who were not doing varsity sports. Now that was a small group, and it's really too small to draw the same type of inferences that we draw from the whole group, but that is what we found in the younger, really more active group as well. So in summary, the LIVE-HCM study showed that in the 1,660 individuals with HCM or gene positive, those who were engaging in vigorous exercise did not have a higher risk of these arrhythmic endpoints compared to those who were less active. Those who were in the high intensity competitive sports were also not at higher risk. And when we excluded the people who were only had the gene, we showed things were very similar. A very important aspect of this study that overall event rates were low in both groups. We had less than 5% experiencing these events over 5 years, and I think this really emphasizes the fact that most of these patients that were in this were followed by high volume HCM centers, which means they had been appropriately treated, they had been appropriately risk assessed. If they needed an ICD, they had one. And I think that was one of the reasons why we had such a low event rate. So the data don't support universal restriction of vigorous exercise in patients with HCM. Rather, the data will inform discussions between patients and physicians regarding vigorous exercise participation. And again, in the context of expert assessment and management, using an individualized shared decision-making discussion. Every person with HCM is different, just like every person with any type of heart problem is different. And this data doesn't say just like everybody go out and do, kind of whatever you want. But what it really says is that that as long as you talk to your doctor, make sure that you've been treated appropriately. If you need an ICD, you've got one, that in that situation, it looks like vigorous exercise does not increase the risk of having these types of arrhythmias. And so again, I'll say that to those who participated in the study, thank you, because it was really, we had a wonderful group of participants in this study, and we do have a long-term extension. So some people may be still participating as well. So to get back to the questions we started with, is it good to exercise? Yes. Is it safe for me to exercise? Generally, yes. And again, it's always important to discuss with your HCM physician and your HCM team. And how much exercise can I or should I do? Well, so for everyone, the HA suggests 150 to 300 minutes per week of moderate intensity exercise. So that's a good minimal goal for all of us. But you may wish to do more. And if so, again, as long as you get there slowly, you don't want to jump from 0 to 60 in 1 second, but as long as you get there slowly, you can do what you want. Again, in conversation with your physicians. And what about high intensity exercise or sports participation? The LIVE HCM results did not show an increased risk of arrhythmias with these sorts of activities. So again, talk to your HCM provider to tailor a sports or exercise program that's right for you. Thank you. So I'll stop and take questions.
Thank you, Rachel. So a couple of meetings ago, we were also talking about exercise in HCM, and a question kind of provoked me to think about things a little bit differently, because we are changing, we are evolving in guidance. And somebody asked, why were they wrong before? And I said they weren't wrong before. What we knew and what we had to work with was limited. We didn't have risk assessment. We didn't have community AEDs. CPR training wasn't as advanced. Emergency action planning wasn't there. Now we have the ability to risk stratify patients, have them consult with experts, protect them, and help them get on living the lives that they want to live, doing the activities they want to do. And most people can do a lot more than we used to think they could do. You like that assessment?
I think in general, as we're just starting to learn about different diseases, different issues, different problems, people are going to be cautious, and they're going to say, in the absence of data, let's be cautious. But I think one of the things that LIVE HCM showed is that people were kind of doing things anyway, and LIVE HCM allowed us to say, people who have made this decision to be more active anyway. Are we now able to get some data? So I think it was appropriate in the absence of data to say we better be cautious. But I think as we've learned more about HCM and have been able to collect this type of data, we're now able to say that a lot of these restrictions really weren't necessary.
I'm going to twist that a little bit and say they may have been necessary years ago, and they're not necessary now.
Okay, yeah, and I think that's
And I think that's a nuance that might have been confusing some people, that why did everything change? Well, the whole world changed. And if you want an example of the change when it comes to athletes and cardiac arrest, you look at what happened to DeMar last January and what happened to Hank Gathers in 1990. And if you watch those two videos side by side, you see what 30 years of work can do to change outcomes in sudden cardiac arrest. So we're in a different time, in a different place. And I thank you for this amazing work and the partnership that we've had through this journey to find better answers. And it's a quest that continues. We do have a question about a specific activity. Was there anybody in LIVE HCM that was participating in weight training?
So that's a good question. And we do have information about the different things that people were involved with. As of now, this type of study generates a lot of data, a lot of information. And at this point, we looked at the sort of the basic primary question, like, can you be vigorous or not? And that's the data that I talked about now. But there are so many other analyses of this data set that we're going to be doing over time. And certainly looking at weightlifting will be a question. I think the question is, if your heart is already thick and now you're going to do something that's going to put more pressure on it, is that going to make it thicker? And I think there's two parts to that question. One is it going to make the heart thicker? But second, is the type of hypertrophy that comes from doing weightlifting, is that a different type of hypertrophy? And if that number changes, does that actually mean something negative is going on? We do have some information that when people stop weightlifting, that their septums or their heart thickness actually comes down a little bit, which tells us that maybe doesn't cause any permanent changes that's like damaging the HCM heart, but it's maybe sort of superimposing the normal types of changes on top. But we don't really know the answer, and it's a great question.
I like the question. So we've had this question before. We're going to have it again. And I'm going to read this one as is, no edits. Do you highly recommend an ICD if patients are exercising, especially teenagers who play sports, or does it depend on the patient?
Totally depends on the patient. So whenever I see young people want to do sports, what we always talk about is let's first talk about the treatment for your HCM, because once we treat your HCM appropriately, then we're going to talk about the sports. So we know in HCM that there's a set of criteria that tell us whether a given person is at risk for having a cardiac arrest or not. And that's regardless of whether they're an athlete. So things that have to do with what their heart actually looks like, how thick is their septum, their clinical history, their family history, what their MRI looks like. So using all of those pieces of information, our risk prediction isn't perfect, but it's pretty good. And we can do pretty well at determining whether or not you need an ICD. We have data now both from LIV and from our ICD sports registry, which came before, that if you have HCM, you have these high risk factors, and you get an ICD, then you're going to be protected when you go back to sports with your ICD. What LIV added was to look at the group of people who were not at high enough risk to say, in those people, can they do sports without an ICD? And what LIV showed, again, we need to look more in more granular detail, but from the data that we have so far, it looks like if you don't have an ICD, then, and compared to people that are active and inactive, the active ones also, again, are not having a higher risk. So we do pretty well with our overall clinical risk prediction as far as whether you need an ICD. And it looks like if you're not at high enough risk for needing an ICD in general, that sports is not going to significantly increase your risk again. But this is a very individual conversation because, again, every person is different. So it's a very important conversation to have with your doctor.
I think I get a question on this at least 3 or 4 times a month. Well, why don't I just get the ICD? Because then I'm protected. And then you have to explain they aren't without risk. They aren't without complications. It gets complicated to not look at it black and white. It's definitely shades of gray.
Yeah.
We have another exercise related question. This person was advised that planks or any exercise where she's bearing down should be avoided. Is that true?
So no, if you have obstruction that is symptomatic and you do the type of exercise that's a plank, like something that like makes you bear down with your abdominal muscles, that can potentially worsen symptoms of obstruction. But if you don't have symptoms of obstruction and you feel fine doing your planks, then there's not a reason not to do them.
Okay. There we go. We have answered all of our exercise questions. I'm sure a couple more will percolate and you'll have one last bite at the apple at the end. So get your questions ready. And now we are going to flip to the topic that I find fascinating in today's world, because 2005, we were all excited that we could do genetic testing. And now we're talking about gene therapies. So Dr. Mani, why don't you talk to us about genetics?
Okay, thank you very much for everybody who stayed so late to listen to our talks. As John mentioned, I'm a cardiologist, but also a geneticist, and my professional life is divided into 2 parts. One is that I run a lab where we make a diagnosis of genetic diseases, and then we model them in animal model to find out how they work. And the other life is that I actually also attend clinics, see patients, and do the genetic diagnosis based on the genetic testing. So the story of finding disease genes goes back to identifying families, as shown here. As you can see here, in this family, the black symbol, circle, and square represent female and male. Those who are not having any dark symbol, they're unaffected. And very beginning, we understood that almost half of the patients are having a kind of disease. Therefore, we call them autosomal dominant, means one normal gene, one abnormal copy of the gene. And therefore, half of the children are going to be affected by just grossly mentioning it. Now, the way to find the disease genes was based on to see what part of the genome, means your DNA, which is in the nucleus, which is kind of the inheritance kind of a code for you, is shared among those who are affected here and not shared by those who are actually not affected. So going by this, you would look for one single gene that obviously going to have a large effect, and it's going to be rare. Basically 1 in 10,000, 100,000 were the initial mutation rate. So we call allele frequency. But actually it's important, even though the effect size is large, we also noticed from the very beginning that there are, the people who have the mutation, they don't have the disease. And this brings it to a complicated discussion of gene-gene interaction, gene-environment interaction, which I don't have the time to get into, but they can skip generations. Now, the initial genetic testing showed that the proteins that involve in contraction, heart contraction, are the major proteins that are mutated, and showing the two of them here, myosin binding protein C3 and myosin heavy chain 7, were the most common ones. The myosin heavy chain 7, as mentioned by Nikhil, is actually a thick filament and causes a thick filament here, which has a globular head. Sorry, what is my mouse? And the another protein, myosin binding protein C3, is a very small one, which actually limits the contractility of this thick filament. Thick filament having this globular head pulls the thin filament acting together and causes contraction. Now, based on having large families, multiple families, there were genes that were established as definitely disease genes. I mentioned 2 of them here, but there are more of them, many of them actually involved in contraction of the heart. Now, this was an initial approach, but recently we have a number of genes that we know that cause heart, HCM. So we can actually do a panel testing without having a family, but just getting the blood samples of the index case, the individual that comes to the clinic, and do a panel. Now, there is an alternative to that testing, and that actually is called whole exome sequencing, in which we actually screen the entire genome, all the genes in the body. And the advantage of is that you may discover things that you never knew about, is a novel gene, with a caveat that you may find a protein that is mutated, that's in the heart, and you think is causing disease, but that's a false positive result. Therefore, we actually examine the extended family and look to see how this actually mutation is segregated and how rare it is and how plausible it is that it's causing damage to the protein. So based on that, there are some genes that we call likely causing, and they are list of it. You don't have to really memorize or know, or etc. Some of you may have been having these mutations, but these are likely. But this can change, and in future, maybe we find out that some of the mutations we thought are actually causing disease be actually benign mutation, which I can explain in next slides. So when you get a genetic testing result, it may come as a pathogenic variant, and that is actually a mutation in a known disease gene, let's say myosin heavy chain 7, thick filament that I mentioned, and that is specific mutation, exact mutation was known to cause disease. Generally, these are altering the protein function or structure. For example, if I say, for example, a gene is a chapter, and the amino acids, the building block of it is a letter, you may see that the book becomes cook, completely different meaning. Lock becomes luck, and completely different meaning. So that's kind of how it works. And there are also some likely pathogenic. Maybe you have less evidence for that, but there are still, both of them are rare in general population. That's one sign. And also both actionable, means that you have to look at the rest of the family, see who has it. There are mutations that are benign, means that change in amino acid didn't cause any change in the structure or function of the protein. For example, if a child writes instead of cat with C, writes with K, still people are going to understand, means that it's not going to change anything in the entire thing. Then it comes variant of unknown significance, many of you have been diagnosed with, and that is actually a mutation in a disease-causing gene. Let's say if the thick filament has a mutation, but the specific mutation, the change that can cause amino acid change, has not been shown in other people. In that case, we look at it in genetic centers, that how common is it, And then we have some algorithms with the computer that say, is it changing the structural function of the protein? How can we predict that? Is it deleterious? Then we look at the member of the families who have the disease and try to see if this actually segregates with the disease. And then finally, we have a genotype negative. It means we didn't find any mutation in any gene that we thought is causing cardiomyopathy. Now, if you look at this pie chart, that how many of mutations are kind of sarcomeric protein, means that contractile protein, etc., titin, about in our hands, about 16% we don't find any mutation. And that's obviously disappointing to many people. But I have to say that the several studies have shown that if you don't have any mutation or no family history, your risk of having events is much lower. So this study was published in Circulation, showed that actually, if compared to the general population, the event is higher if you have a mutation based on the age. But this fault decreases if you don't have the mutation or family history. So it's not a bad kind of thing not to find any mutation. I mean, seeing the positive side of it. On the other spectrum is that some people come with more than one mutation, which makes our task very difficult because we don't know really which of these actually is more causing disease. And if a child inherits one of the two, is that child in danger or not? So they have to do a lot of calculations, which is complicated. But in general, more than one mutation has been shown. For example, this group by Ackerman, led by Ackerman, found out actually if you have more than one mutation, you have more at risk of some event. Now, genetic testing, as I mentioned to you, can come with VUS, or you can have a likely pathogenic variant. If you have a likely pathogenic variant, as I told you, you do a cascade screening in the family to see who else is having this mutation. And if it's affected, obviously, you have to do regular follow-up. But if it's negative, and you're definitely sure that this is a disease-causing gene, you don't have to follow up the rest of the family. So they don't have to go do serial echoes and MRIs and whatnot. But one thing I have to emphasize is that we actually have to do regular reevaluation, because sometimes the things we thought is actually are likely pathogenic end up to be actually benign by more having databases, having more patients with those mutations, etc. Obviously, if it's actually your VUS classified as likely pathogenic, you go back again and do cascade screening. It is actually the fact that more of the pathogenic become less pathogenic than the non-pathogenic become pathogenic. But this have to do the evaluation every few years. This is very important. And it's done actually by vendors on one side, and here at Yale by clinicians and the laboratories together. Now, in general, there are not mutations that are worse or better or less severe or more severe, with very few exceptions. Some mutations, like these two mutations here in myosin heavy chain 7, occur in the childhood because they are more severe. The mechanism in these is actually that those globular head of the thick filament, the one that actually slides the actin, are hyperactive. So they are hypercontractile. And that's why the disease comes in their childhood. In contrast, actually, in adults, is a little different. Normally, as Nikhil explained to you, you don't actually use all of the myosin heavy chain. In general, about 50% is sufficient to be used. And what happens is that in the patients, actually in adults, they generally, the other head that has to be relaxed can become actually active, and both of them can be involved in a sliding of actin. So if the disease is hyperactivity, here in this case is actually more myosin heavy chains getting involved. So normally half of the myosin heads are sufficient. In adults, they actually become more engaged. More heads become engaged. As a result, if you look at this curve, looking at the force, the mutations in general, what they do, they increase the calcium sensitivity, they increase the enzymatic activity, we call it ATPase, which is breaking down your energy source, and less relaxation state. Now, one person asked about testosterone. Testosterone does actually similar things. So therefore, should not increase the side effect of the drug that you're going to reduce contractility. That's at least you can make an inference. So in general, the mutations in myosin heavy chain 7 are gain of function. They may more active. and therefore they are not truncating. They are not missing one allele. They are mainly what we call a missense mutation. And they predict they are mostly localized in the head of the myosin and in the neck, which they have to relax, sit on each other, and they do not relax, and the head becomes engaged. Myosin binding protein C3, which have to limit the function of myosin heavy chain 7, generally are loss of function. They either you miss one allele because one protein is mutated, that it truncates the protein, or it is a missense mutation, means that amino acid change that reduces the function of the protein. And then finally, because of this mechanism that I explained to you, the drugs were developed, which I'm not going to get into it because it was explained before, to reduce the function of the contractility. And, main effect obviously is LVOT gradient, improves the signal, the symptom score, and as they mentioned, it made it drop actually ejection fraction by 10% in one study. And luckily, it's actually in most cases reversible if you kind of screen the patient with echo that was explained. Now, do we have any gene therapy? So there have been actually attempts, and there are different techniques, mechanisms. Recently, you guys have heard of as CRISPR, and that CRISPR means you either change a nucleotide, means a kind of building block of their DNA, or you actually remove the entire gene. Both of them have been used. People have used actually 2 different viruses to get that kind of a CRISPR to the heart. And in one case, they completely change that building block, that the nucleotide. In another case, actually, they go after the copy that is abnormal, remove the whole gene. And there have been limitations. One is that you don't get to all the cells with that virus. Still, work is being done to actually target almost all the cells. Another problem is that actually you can target the wrong gene. And I'm sorry, my mouse is moving. You can target, so off-target effects that obviously then is going to be reducing on contractility, things that actually mavacamten can do with actually more severe kind of consequences. So there is work being done to actually improve this, and hopefully we're going to have, by using multiple viruses and also improving the technique, using a gene therapy. So if I summarize what I said, all patients with hypertrophic cardiomyopathy should undergo genetic testing, and preferably at specialized centers where we have, for example, Sarah, who's a genetic counselor who can explain to you all the details, false positive, false negative, how to bring the family members. Patients should be informed of the false positive, false negative results. The cascade screening should happen. Family members with a pathogenic or likely pathogenic variant should be screened for the mutation. Unfortunately, the VUS, when diagnosed, and even if I think that VUS may be causal, many of the insurance companies do not approve that we do cascade screening. We had some mechanisms to actually screen this patient in the past. Hopefully, we're going to reinitiate that. There was some funding issues. We used to do actually the whole genome screening, and hopefully we can do that. But at this moment, the insurance companies do not approve that VUS is being used for screening of the rest of the family. Genetic variants should be evaluated every 2 to 3 years. If you have a VUS, ask your doctor in 2 years or 3 years, did the VUS become pathogenic, or is it still, or it became a benign mutation? This is something that you have to interact with your physician to make sure that is done. And patient with no genetic diagnosis doesn't mean they don't have the disease. They just have to be followed clinically, because in about 15 to 20% of the patient, we don't know what's the disease-causing gene. Now, I don't have time to explain to you that there are also mutations that actually are not in the protein coding region. They are what we call it in the intron, intergenic, and they affect the expression levels of these proteins, but they have very small effects, and multiple of them are necessary to cause disease. At this moment, we do not do genetic testing for those, because as you can imagine, when you have multiple mutations, your children are not going to inherit all of what variants that you have. They inherit probably half of it. So there is no consequence for those cascade screening. And once you have the disease, actually, there's not much value to know if you have a mutation in the genes that has very small effect. So thank you very much for your attention. And if you have a question, I answer you.
So I'm going to start you off with a question that you didn't talk about, but we're getting questions about it now. Some of the companies are now offering a genome evaluation rather than a panel.
Yes.
Can you talk a little bit about the difference between the panels and a exome sequencing?
Yeah. So actually, we started here as the first center in the country to do the whole exome sequencing, not whole genome. The difference is that whole exome is that you look at the part of the genome that actually encodes protein, while in whole genome, you look at all other regions, the entire 5 billion base pair. And yes, the vendors actually are now doing that because for some of them, they have established a system that is cheaper for them instead of going after all kind of, 30, 40 genes. And they can offer you. The difference is that we used to actually evaluate ourselves, look at it, and do our interpretation based on our knowledge. The vendors give you a results that they found or they didn't find, but they haven't provided yet the entire results to us to look at it and be able to maybe guess. For example, there are a lot of proteins that are expressed in the heart, right? And I name one, NFAT2, for example, that is involved in actually testosterone-mediated calcium signaling and hypertrophy. And if I find somebody have mutation in that, I may say, wow, look at that. I mean, this is a rare mutation. It may cause disease. That result we don't get from the vendors yet. We are actually negotiating with them. But yes, that is absolutely right. They do it, but what they're going to use for the interpretation, they're going to look at what genes are known, and they say, okay, you had mutation or you didn't have mutation. The end result is not much different than doing a panel. But we had this here, and we had a kind of a center called generation. We used to actually get the DNA. We used to look at all the coding region. It's very complicated to do whole genome, by the way, because you're going to find so many variants that it's difficult to interpret. And then we used to say, okay, there are some genes that are mutated in genes that were not known to cause disease, but we assume it's possible that it actually caused disease. And then do you have any family members who has that disease? And then we would bring them and test them for that mutation and say, wow, everybody who is affected in the family has this mutation. Everybody who's unaffected doesn't have it. And say this very highly plausible that this is a disease-causing gene. Did I answer the question?
You did. It's a complicated question because we've programmed everybody to think about gene testing, and now we're going, okay, now we've added a layer to it, and what does that mean? And I think we're all still trying to figure that one out. I've met with a couple companies who are offering this. I'm like, how are we doing it? So I'm talking about the operational side and the reporting right now, and we'll
We are in
hopefully
negotiation with Invitae to give us the results, and I'm sure Sarah is going to work with me on that, and we're going to do a lot of research there.
Absolutely. So what I thought we would do is we would shift to Sarah for a minute because we do have a couple of genetics questions, but they might be answered in what Sarah has to offer, and then we'll go back and we'll talk a little bit more about gene questions. Sarah, off to you.
All right, sounds good. So I'll share the screen. Okay, there we go. So I know we're a little bit behind schedule, so I'll try to be prompt here. But hi everyone, my name is Sarah. I'm the genetic counselor who works in the inherited cardiomyopathies program here at Yale and YNHH. So first, I know from the poll that a good proportion of the participants on this call had undergone genetic testing for HCM. So many people might already be familiar with this, but for those who aren't, I get a lot of questions. What is genetic counseling? What do you actually do? So briefly, genetic counseling is really just the process of helping individuals and sometimes their family members as well understand and adapt to a diagnosis, help them learn what their options are for genetic testing, and learn if testing is something that, works for them and makes sense for them. And then additionally, to help them figure out and understand, what are my results and what do these mean for me in the context of my diagnosis or family history. In addition to that, we often wear many hats. Of course, there can be issues with insurance. Communicating results between family members can sometimes be a bit tricky. Sometimes these patients are going through really difficult or emotionally stressful situations. So often providing some emotional support or guidance is part of the job. Also things like helping, with patient advocacy and occasionally doing some research work as well can be part of the job. And while I primarily work in inherited cardiovascular disease and cardiomyopathies, genetic counselors, as you probably know, work in a lot of different areas such as prenatal genetic testing, cancer genetics, etc. So just to kind of give a bit more detail about what a typical genetic counseling visit might entail. A big chunk of it is assessing the patient's, the diagnosis, the personal history, the symptoms they may be having, as well as their family history. So a pedigree, we've seen these images before down here. It's basically a family tree, where we can delineate people's diagnoses, whether they've had genetic testing, who's deceased, who's still living, if a patient has children. So that's a way to kind of lay out all the history kind of neatly in one chart. Often there's a lot of education about, the genetics of the condition, how it may be passed down to children. Of course, what are the options for genetic testing, how the genetic results could potentially, impact maybe treatment or a management plan for patients. Of course, genetics is a family affair. So what are the implications for family members potentially of that testing? And then finally, there's often a good bit of discussion about, logistical considerations or insurance concerns, namely, is someone's insurance going to cover a test? Will I have trouble getting life insurance potentially if they see that I have a genetic diagnosis? So those are often things that will generally come up in a session, whether it's focused on HCM or something else. So now more specifically, talking about genetic counseling and potentially testing for HCM. As you can see, I am just one part of a large team here, working with a lot of other specialists. Generally, the way that I see patients is that one of these doctors on the call will refer them to me after seeing them. Less frequently, sometimes we may get referrals from outside cardiologists or even other genetic professionals within Yale. For example, maybe the cancer genetics team is seeing someone and they end up uncovering a family history of HCM. So we do sometimes get cross referrals in that way. So for HCM, genetic testing is utilized alongside clinical tests like echo, EKG, cardiac MRI, really as part of the diagnostic workup. It's definitely just sort of one piece of the puzzle here. And Dr. Mani did a really great overview of the, the genetics of HCM and how the genetic results can often come back in a couple of different ways. So to give a kind of really brief summary of that, results can come back positive with pathogenic variants, essentially meaning, we've identified the genetic cause of the HCM. They can come back negative, meaning we didn't find anything, or they can come back with a variant of uncertain significance, which of course means we're not quite sure if this is exactly the cause of the individual's diagnosis or not. So a big part of this process is often, how do we deal with this uncertainty, right? What does that mean for the patient? What does that maybe mean for their family members? And of course, there's still a lot we have yet to learn, not just about the genetics of HCM, but genetics in general. So as mentioned prior, just because we aren't able to find a genetic result for a patient, even if the results are coming back negative, it doesn't mean that there isn't a genetic basis. It just means that maybe it's something that we're unable to pick up on, with the genes that we currently know about or just our current testing technology. Now, something to note for HCM specifically, the genetic testing doesn't really change the course of treatment or, drastically change the patient's management or anything like that. It's more of a way of getting more just information about the cause of the condition, and for us to learn, how do we best manage the patient's family members? Are they potentially at risk? And then another important aspect, for the genetic counseling and testing is the access to testing. So something that we do is, not just in-person visits, but also visits via telehealth. We're able to send specimen collection kits to patients' homes so they don't necessarily have to, drive out to the lab and get lab work done. So that's something that's been really helpful, especially, in light of COVID. In just improving access to the genetic counseling and testing process. So Ari and I must have been thinking along the same lines, because we both, without discussing it, included the same graphic from the American Heart Association. So I won't, belabor the point here too much, because we already went through this a little bit. But I just like how it shows that even though genetic results can sometimes be a little bit confusing when they come back, they can be kind of uncertain. We do have expert guidance on, how do we utilize these results, on how to best manage the patient and their family. As mentioned before, if we do get a positive result, if we're able to identify a pathogenic variant, then we're able to do cascade testing and screening for other members of the family to learn, who else has this mutation, and who else may be at risk. And if someone does not have any actionable findings, if it's negative or perhaps an uncertain finding, then we know that the next best step is to do clinical surveillance, to screen people with echoes and EKGs, basically to check and see if anyone else in the family might have any manifestations of HCM. So like I said, genetics is really something that can involve the whole family. As many people probably know, the inheritance of these mutations is typically autosomal dominant, meaning that if, a patient has an identified mutation, there's a 50% chance for each of their first-degree relatives, so parents, siblings, children, to also test positive for that same mutation. So obviously this is something that can be really impactful for the individual's family members, and again, it's, the processes here can either be doing targeted genetic testing and then follow up if necessary, or clinical cardiac screening if there aren't any actionable genetic findings. And of course, this is something that can be really stressful to talk to your family members about. So often part of the genetic counseling process is, okay, how are we going to effectively and appropriately communicate this information to the relatives, to people who may be at risk? So I've had many conversations with people about how to broach the subject, how to talk about this topic with, younger kids or children. And something we offer is basically a family letter, where we can kind of write out and explain all the information. And then it's really just on the patient to, perhaps drop the letter in the mail or send an email or something like that. And we can really take care of, communicating what the situation is, and here's the information you should know. And then finally, sometimes we may also have discussions, for individuals with a positive genetic result who are thinking about, having children and planning out their family. So something that ends up being an option for some people with these identified findings is a process called IVF with preimplantation genetic diagnosis. And it's something that's not an option for every individual, but essentially it's a way to conceive a child via IVF, and genetic testing to ensure that the embryo does not have the HCM mutation. So it's a way to have a child, guaranteeing that you do not pass down that specific mutation for HCM. So as you can see, often there's a lot of separate discussions that kind of go along with the process of genetic counseling. It can be sometimes pretty basic and to the point, and sometimes there are many other considerations that go along with it. So in conclusion, the genetic counseling and sometimes testing process as well can end up being a helpful source of information, not just for the HCM patient, but their relatives as well. And I've just included this link here about geneticcounselors.com. It's very aptly named. So that's a site by our professional society, NSGC. And there's also a pretty helpful tool on there. So if you are interested in not just necessarily for cardiology, but any type of genetic counseling, there's a way to find a genetic counselor near you. And finally, that's my email address there. So if anyone has any questions after the fact, they can feel free to reach out to me. Right, now I'll stop
Thank you,
sharing.
Sarah. I have a couple of genetic questions that have piled up, so we'll go through those real fast. While I am doing this, I'm actually going to launch a second poll that you'll have a minute to think about, and it is simply, audience, what topics would you like to hear in future Bighearted Warrior tours? So if something pops into your mind, just populate it for us. We appreciate your feedback. Okay, so perhaps this has been addressed already. If my results were negative, no specific gene was identified, how should I push my kids to get screened? Clinically, echo, EKG, checkup with cardiologist, and revisit genetic testing every 3 or so years.
Yeah, definitely. That's what I would recommend. You know, if you had testing done in the past few years with a comprehensive panel and it was negative, then your children pursuing genetic testing isn't really going to be helpful. The best way for, us to keep an eye on them would be for them to get screened clinically. And then of course, over the years, the labs expand on these panels and just the technology improves. So certainly we've had people where, maybe they had testing done like 8-10 years ago and their results were negative. They've come back and say, hey, let me do testing again. And we've found a result for them that time. Yeah, so definitely in that case, the children should be screened clinically.
Another question, similar yet different. How is a negative different than an uncertain significance?
Yeah, so basically a negative result would mean that the lab didn't identify just any changes within any of the genes that they're looking at. And basically you can take a VUS to mean that the lab saw some type of change that they detected within a gene, but they just don't have enough information at this point in time to put it in the camp of, this is definitely pathogenic and associated with the disease, or this is totally benign and doesn't cause any health problems at all. And it can be kind of tricky because humans just have so much genetic variation that's possible amongst ourselves. And many of these changes actually don't cause us any issues and they kind of just make us unique. So that's kind of how they differ. And like Dr. Mani had mentioned earlier, with the VUSs, sometimes there's a bit more research that can be done into the particular finding. There are computer models that we can use to predict the effect of a genetic change on the protein. Sometimes genetic changes may have been reported in the literature before. Occasionally it might be helpful, if there are other affected people in the family, to do some further testing and investigation of the VUS. So that's something that's more so done on a case-by-case basis. But also, like Dr. Mani mentioned, it's important to kind of follow up on these things maybe every few years, because the labs, the commercial labs do gather more data and they are able to eventually reclassify the VUSs as being either pathogenic or benign. So usually when I have VUSs, I say, just because we don't have enough information on it now doesn't mean that in the next couple of years, we're not going to get more.
Okay, so now the complicated one. Dr. Mani, you're off mute. Good.
I saw that question.
If you have both myosin heavy chain and myosin binding protein C, is your presentation different as they seemingly have opposite interaction?
Right. So I think there was a misunderstanding here, and that is that their natural function, the physiological function, is opposite. So one puts brake on the other. So myosin binding protein C3 puts a brake on your myosin heavy chain 7. That's a normal condition. However, the mutation in myosin binding protein C3 takes off that brake, and therefore myosin heavy chain 7 can become more active. And the mutation in myosin heavy chain 7 makes it even more active. So I practically probably showed too much data or went fast over it. If you have more than one mutation, generally, I mean, look, there is no rocket science here. There is not a black and white. There is no yes or no. But possibility is that actually potentiates the effect of each other and is actually worse to have that. Becomes complicated then if you look at your children and one has one and the other one has the other one, or which one is really real doing it. And that is what we actually, as John Stanel mentioned, this is a topic of our discussions on our meetings, to go over possibility of which one actually really causes disease. And then we go into that, how rare that variant was, how much predicted by, as Sarah said, on your using algorithm, how it can change, and make a guess. Right now, actually, because I'm a reviewer on the grants, a lot of people are trying to use high throughput methods to actually induce these mutations in vitro. And whether this is iPSC using or whether there's actually myosite, to predict and then do entire kind of amino acid substitutions and to see which one is causing. Actually, this is ongoing, and I'm sure the data is going to come out within a few years, and we're going to have more data. But then you're going to have fewer VUS. They're going to be more kind of understanding of which one is pathogenic. But at this moment, is like kind of guessing and based on all this data that I told you. Let's say, for example, if your myosin binding potency 3 is 1 in 100 in the population, and the disease is never 1 in 100 population, you can say, well, that may have a small effect, but it's not really that major effect. That's, for example. Or if amino acid is changing within the amino acids, which are the building block of proteins, that have different groups. And if the changes with the same group with a little bit tiny bit of a changes in, small changes, we say, look, this is not going to really cause a major defect in your protein. So that is my answer. So they can potentiate each other. That potentiation may be small or may be significant.
Okay, we are running very late tonight, so I'm going to hold the rest of these questions until after our last speaker, and then we will do rapid fire question and answer. So I'm going to ask our panelists to read the questions that are in queue, with the exception of the top anonymous comment, and get ready for speed round after Dr. Sen speaks. You're up. There you go. Beautifully done. Thank you. Oh. It's a Mac.
We're good?
We're good.
Thanks, everyone, for hanging in there tonight. In the last few minutes, I will kind of talk about how to manage advanced heart failure and hypertrophic cardiomyopathy. So I'll start with the take-home points. And I think I'll come back to this at the end, but my goals are to kind of demonstrate that advanced heart failure can be insidious in hypertrophic cardiomyopathy. Hold on one second. Are you guys seeing my screen here or no?
Yes, we do.
Okay, perfect.
I'm seeing the screen, but we're on their first slide.
Oh, I'm sorry about that. I don't know why that's happening. Give me one second.
Technical difficulties, please stand by.
Always on the last talk too, right?
That's okay. We'll unshare, reshare.
There we go. So yeah, I'll start with the take-home points, and that is that advanced heart failure can be quite insidious in hypertrophic cardiomyopathy. And one of the insidious elements of advanced heart failure is the development of high pressures in the lungs, known as pulmonary hypertension, which can make treatment with heart transplantation, which is the gold standard for heart replacement therapy, quite challenging and sometimes even not possible. And I wanted to highlight some of the kind of changes in the transplant allocation system that we use in heart transplantation, with specific considerations to enable improved access to transplant for patients with advanced heart failure with hypertrophic cardiomyopathy. And finally, highlight some of the newer methods in transplantation to expand the donor pool that have really improved access to this therapy for all patients, including those with hypertrophic cardiomyopathy. So advanced heart failure in HCM is actually a clinically distinct state from other forms of advanced heart failure. The progression to predominantly non-obstructive end-stage or quote-unquote burnt-out HCM has often received less recognition. And I think we're seeing this more given reductions in sudden cardiac death, improved longevity in individuals with HCM. And it's estimated to be around 1 in 15 or 5 to 7% of individuals who have HCM. And it can present in many varied ways. And this is just one example of one of my patients who had significant pump dysfunction. You can tell here on the echo that the left ventricle, which is still quite thick, but doesn't really pump very well, and decreased squeeze or ejection fraction, as we call it, and progressive stiffening of the heart. In other cases, this is a patient, actually a child of a patient of mine who underwent transplantation, who had a very significant thickening of the heart, quote unquote normal pump function or mild dysfunction, but very, abnormal stiffening of the heart given how thick it was, and advanced diastolic dysfunction. And then the third kind of phenotype that we notice is somewhere in between. And so I think understanding that the presentation can vary, and in individuals with hypertrophic cardiomyopathy, oftentimes they're younger than our typical transplant recipients. 50% are less than 50 years of age. They may have mild or moderate pump dysfunction or systolic dysfunction, but more commonly, they have very debilitating symptoms of progressive shortness of breath, inability to be functional and live their lives the way they want. And given the kind of difficulty in how individuals will present, I think it's important that we take like a multi-pronged approach to ensure that we're able to detect signs of progressive heart failure. And we know our patients are big-hearted warriors and have often been dealing with this diagnosis for years to decades. So just asking questions is the start, but sometimes not enough to really tease out those who are at higher risk. Certainly imaging, the pictures I showed of echoes and MRIs can be helpful to detect worsening heart function or signs of increased stiffness. And sometimes it's not enough. I oftentimes rely on other non-invasive testing, such as cardiopulmonary exercise testing, which can help delineate how well the body is able to extract oxygen during exercise, and particularly in patients who feel okay at rest but have a lot of symptoms with exercise. This can be a useful tool to kind of understand how well the body is extracting oxygen during the exercise. I think it's important to acknowledge that given the younger age of presentation in individuals with HCM, we must be mindful of not just using absolute cutoffs in using this testing that we oftentimes do in other individuals with advanced heart failure, but looking at kind of normalized or percent predicted measurements relative to their age, I think is quite important. And finally, invasive assessment of measuring the pressures in the heart and looking at the actual cardiac output can be useful. And then specifically understanding if there's any development of pulmonary hypertension. And pulmonary hypertension is actually a silent marker of end organ dysfunction in individuals with heart failure in general, and particularly those with hypertrophic cardiomyopathy. And the premise behind this is that as the heart gets stiffer, the pressures in the left ventricle, shown here, can get elevated. And this reflects back into the top chamber, the left atrium, and eventually into the blood vessels that feed the lungs. And over time, just as we have a high blood pressure, we measure blood pressure in arms at clinic visits, there's also blood pressure in the lungs. And over time, this blood pressure in the lungs can get elevated and sometimes can be fixed. And that can make transplantation eventually kind of challenging. You can imagine when the new heart is sewn in, the right side of the heart has to kind of work against this high pressure, and if it's fixed, it's been associated with kind of worse outcomes after transplantation. So I think acknowledging pulmonary hypertension, having respect for pulmonary hypertension in HCM is critically important. As I mentioned before, heart transplantation is the gold standard for heart replacement therapy in those with advanced heart failure with HCM. And indeed, in the last kind of phase of transplant in the country, the proportion of heart transplants in HCM has increased from 1% to around 3.5%. As I mentioned, often younger recipients, and the long-term survival is excellent. It's analogous and sometimes even better than other individuals who are undergoing transplant. And certainly there can be some early post-transplant challenges, and oftentimes this is felt to be related to other comorbidities, but in hypertrophic cardiomyopathy individuals, oftentimes there are not that many other comorbidities, but pre-existing pulmonary hypertension emerges as an important challenge. In the US, we perform around 4,000 transplants a year. Median survival is 13 years. That's among all comers. And that's different based on age of the recipient as well as the comorbidity burden. And the way that we allocate hearts for individuals who need transplant has changed in 2018. And this was, in the previous era, there was a 3-tiered system where individuals were listed for heart transplantation. They kind of fell into 3 buckets: people who are supremely sick on mechanical support devices or on IV medications, and then everyone else was kind of lumped together as a status 2. In 2018, in October 2018, this changed to a 6-tiered system, which was attempted, the intention was to kind of have better granularity at the waitlist risk or waitlist mortality of certain individuals, and try to allocate hearts based on that risk. And I think a lot of credit has to be given to the thoracic committee, as well as to, I think, Lisa and her team were important champions of having hypertrophic cardiomyopathy kind of fall into a different category in and of itself, acknowledging that it's a unique state with different risks compared to other individuals. And I think that's a really important change in the allocation system for transplant. And to dive into it a little bit in more detail, recognizing that the unique presentation in HCM can be associated with increased waitlist mortality and oftentimes a narrower window for safe transplant. And individuals with HCM oftentimes can develop instability different from other folks who have advanced heart failure, and irreversible pulmonary hypertension becomes a key challenge. And specifically for HCM or other restrictive cardiomyopathies, there is an alternative exception pathway. So as I showed in the previous slide, individuals with HCM who are listed for transplants are also oftentimes start at so-called status 4. But based on their pressures in the heart and other factors, their status can change, because this suggests that their waitlist risk is higher. And there are specific criteria for status 3 and status 2, specifically for individuals with HCM. And I think this is very important to acknowledge. I'd say early on, after the transplant allocation system changed, I don't think this was as widely recognized across the country as it is now. And I'm happy that it is now, because I think it recognizes that these individuals have separate risk profile than other patients waiting for transplant. And it's important for us to be aware of as a community. And finally, I'd like to kind of talk a little bit about how we use new technologies to expand access to transplantation, and specifically talk about a method called donation after circulatory death. So, historically, I guess the first heart transplant in 1967 in the world in South Africa was done by Dr. Barnard, and the donor and the recipient were kind of in the same or adjacent operating rooms. And the donor was not brain dead. I think had a motor vehicle accident, unfortunately. And then the heart was taken out and placed into the recipient. As you can imagine, operationally, this can be a big challenge. So this idea of co-locating the donor and recipient kind of could lead to restricted use of transplantation across the world and across the country. And so eventually, the majority of donors are kind of legally declared brain dead. And then these hearts can be assessed functionally in the donor body. It's a beating. And then once it's taken out quickly, it can be stored in ice and transported 3-4 hours, maybe a little bit more, and implanted into the recipient. Unfortunately, we have a huge shortage of donor organs, but fortunately, we've had development of technologies that have allowed us to perfuse the heart outside of the body. And there's been this renewed interest in looking at or utilizing donors who have not met brain death criteria, but do have neurological criteria for kind of no viable recovery, and with the family decision to withdraw life-saving treatment. And this has led to this idea of donation after circulatory death. In this method, the donors were appropriately identified, life support is withdrawn, you wait for specific metrics of the blood pressure being low or the oxygen being low, and the heart is actually allowed to stop naturally. There's a standoff period where nothing happens, and then the individual who has donated the organs to society is confirmed as dead. And then the organs, in this case, the heart, is taken out. And this has to happen within a short period of time, oftentimes 30 minutes or thereabouts. And then the heart of the donor is placed on this machine called an organ care system in the United States. And it's beating. We can assess the heart visually. We can check labs on it. And this is very different from the typical cold storage that we use for brain dead donors. And then it's kind of transported on this machine called the organ care system, and it can be transported back to where the recipient is waiting. This has really revolutionized our access to donor organs. Just for our center in a small state of Connecticut, typically we were always kind of confined to this radius of like around here. As you can see, we've been able to get out to the West Coast a little bit more, and this has really helped our patients who are awaiting transplant, including those with hypertrophic cardiomyopathy. I'll end with an example of one of my patients, and this was, I had permission to share this, and this was publicly available in the news, but this was a young patient of mine who was in his 30s who had advanced heart failure from hypertrophic cardiomyopathy. Unfortunately, he was seen. He was quite sick, and he was actually sent home on hospice from another place that he was being cared for, and he arrived to us incredibly sick. We were able to kind of help him stabilize on a temporary, or actually a durable heart pump on left ventricular assist device, and kind of get him stronger and better. And then he ultimately underwent transplant using a DCD as a method, and he's back to life. And he's back to work, and is living has new leg in life. So I wanted to share that story as a kind of a culmination of all the things that we've done in the community and in society for our patients with HCM. So I'll end with the take-home points again. So advanced heart failure can be insidious in HCM. But we have to have respect for pulmonary hypertension. The allocation system has changed. There's more work to be done, but it's important changes for HCM specifically. And newer methods to expand the donor pool have really revolutionized transplantation in 2023, 2024. Thank you very much.
Thank you very much for your presentation. And I want to remind everybody how good transplant outcomes could be in HCM by giving a little history lesson. In 1959, Eugene Brunwald diagnosed the very first patient with hypertrophic cardiomyopathy. His name was Claude Brady. And I'm very happy to say Claude was a friend of mine for many, many years. Claude had a heart transplant in 1988. And he lived until January of 22, where he died at the age of 84 from kidney failure. His heart was in beautiful shape. So he was an early transplant. He was only in his 40s when he was transplanted, and he made it to 84. If you ever hear me chant, "Be like Claude," this is my rally cry to all my transplant buddies out there, or people who are thinking of transplant. I aspire to be Claude Brady. I aspire to acquire 84 years on this earth with my donor heart, and it is possible. I've seen it happen multiple times with other individuals, but HCM does really well long-term with transplants. So be like Claude, everybody. All right, boys and girls, we are in the final chapter. So we are going to go to rapid fire question and answer. If I'm not my sparkling, typical, like bubbly self today, I was at a dinner meeting until 10 o'clock last night. I was up at 7 o'clock in a meeting in South Jersey, drove back to North Jersey, landed at my desk right as this was starting. I'm just a little tired today. With a donor heart and everything, you still get tired. Just remind you people with HCM that's the way it happens. And yes, the HCMA did participate in very aggressive advocacy on behalf of making sure patients are listed by disease type with a classification so that we had an entry point that was consistent around the country, because different centers used to view HCM quite differently. So we got some standardization. Now we work on improving that. Okay. Where am I going to start? I'm going to go back to the genetics. There's a couple of genetics things that I wanted to get to, I think, that we didn't. Do genetic labs routinely reevaluate VUSs on genetic testing, or do you need to be retested? How often should these VUSs be reevaluated? And I'm going to start the conversation by saying I was part of an NIH grant that looked at the question, what do we do about VUSs? And there were bioethicists, there were lawyers, there were clinicians, there were genetic counselors. Everybody came to the table. The labs were there, and they all had problems with the system because we just created it in like 2005, right? Of how do you go through this repetitive system? And there was a lot of questions that were brought to the table that need answers. Who's responsible for the relationship? Is it transactional between the doctor and the patient who did the gene test? Or is it a relationship that lasts longer? Who's going to pay for the storage and reanalysis of this information? What are the legal obligations? What are the regulatory obligations? And there weren't a lot of great answers, but a lot of really smart people are looking into each of the subsections. There will be a couple papers coming out in the next couple of years on this, but it's evolving. Dr. Mana, Sarah, any comments on VUSs and reanalysis?
Sarah, you want to answer or you want me to?
Yeah, I mean, I can say, you know, like Dr. Mani emphasized earlier, it's important to keep that conversation going with the provider. You know, personally, at least I can speak from my experience working with these labs. Usually it's a couple of years before we get updated information. And at least some of the labs do reissue new reports, you know, directly to us that then we're able to share with the patients regarding this. So usually I would say if it's, you know, been within the last few years, then there's probably not a need to be retested unless it's been more like on the scale of maybe like 5 to 10 years, where, you know, there may have been actually new genes added to the panels. Maybe the technology has just improved. And in those cases, we would usually encourage, you know, maybe retesting. But yeah, generally speaking, I would say, you know, I would recommend maybe once a year or so, just kind of checking in with the providers saying, you know, have there been any updates? And there may not have been, but it's definitely good to keep that conversation going, especially Lisa, with what you just mentioned. We're kind of figuring out the best way to run these things, like who's responsible for what exactly. You know, so I would always say just generally it's in the best interest of the patient to kind of advocate for themselves in that way and just at least make sure that that conversation stays open. Yeah, Arya, I don't know if you'd add anything to that.
Well, yeah, I mean, exactly what you said is correct. The only thing I want to mention is that any of you who have had the testing not by vendor, for example, with our own West Campus Center, and in that case, stay in touch and kind of we have to communicate with us and we have to kind of talk to the lab and re-examine this thing. In general, even exome sequencing, which means the whole kind of all the genes that we did, technology has improved. So there were times that we missed some mutations. If it is actually even old for that technique is also, we do recommend the repeating of testing. The vendors, as Sarah said, most of the time they actually readdress that. And yes, the panel hasn't changed in the last few years, the number of genes and the genes that were definitely causal. But as Sarah said, over 10 years, you have had evolution of all these genetic discoveries, and we repeat that. We actually repeat that as if the DNA is available, we go run it on the DNA. If not, we draw again the DNA. So it depends on how it is. But the important is, let's say a lab, NVIDIA sends me that this has become a VUS. I have to have a contact with that patient to just kind of communicate that. That now became pathological, or no, it's actually not pathological. It's actually rather benign. So the patients has to be an advocate for themselves.
Sticking with genetics, if an individual has a gene variant, I'm assuming they're calling it a pathogenic mutation, is my guess, but neither sibling has it, is it to assume that the genetic mutation started with that individual? Or could it have come from upstream? If it's the siblings are clear, what about the parents?
Both scenarios are possible. Let me explain to you that we have what we call de novo mutation. That can be only discovered by a trio testing. Many actually of these vendors suggest trio testing, which means parents and the child. In that case, if the parents are unaffected, means that they are not carriers, let's say, of the mutation, then it's actually de novo. We're starting with that patient. However, there's also a possibility that the parents are unaffected. As I said, is you can skip generation. But the only way to identify this is by trio testing, simply said.
So just to clarify that a little bit, because this gets a little confusing for people. Incomplete penetrance versus skipping. Can you talk on that for a moment?
Well, it is the same thing. I mean, when we say incomplete penetrance, means that you have the mutation, you don't have the phenotype. And there are many reasons for that. It's rarely known, actually, in the lab. Occasionally, we find that there's another mutation that was beneficial, kind of compensating for that effect of the other mutation. But in most cases, we don't know. And, you can have even kind of identical to twins that they have the same mutation. One is actually having cardiomyopathy, the other one doesn't. And that tells you that there is some environmental effect, etc. And so therefore, that is called either skipping, because it means that the person didn't have it, or incomplete penetrance. There's no difference.
Thank you.
Lisa, can I just also clarify, for if your parent has a positive gene, each child has a 50-50 chance. So it's kind of like boys and girls. So like if one, you could have 4 children, like if, they could be 2 boys and 2 girls, but they could be 4 of one or 4 of the other. So it's certainly possible that if you have 3 siblings, that one might have it, 2 might not. 3 could have it, 3 could not have it. So any combination would be possible. So as Ari said, you really don't know. It's possible that a parent had it. It's possible that it was de novo.
Thanks for the clarification. I have a clinical trial type question. If you read this one, for HCM patients involved in clinical exercise trial, at what BNP level should the patient question continuing the activity or seek independent, not tied to the study, assessment of the risk of continuing in the trial, presumed normal BNP pre-trial level? So number one, I'm unaware of any current clinical trials that are using BNP biomarkers to evaluate exercise. Am I missing something on that one? Is there any trial out there using BNP for an exercise trial? For the myosin inhibitor trials, they're monitoring these biomarkers. So, Doug, I'm not sure which trial you're talking about. Does anybody else have any ideas here? No? Okay. Doug, call the office. Happy to talk it out with you and get more details if you're still here. Okay, so next question. Given the mental health issues that come from having HCM, and there's a lot of them, and we're working on some programs there too, and the fear of exercise along with it, which many of us who were diagnosed back in the day in the 80s, 90s, and were told exercise was not good for us, we have a psychological trigger that it's bad. So we're all getting over that together, and we're going to learn how to exercise together. Why is cardiac rehab not easy to qualify for? And this is an excellent question.
That one?
Yes,
That is
please.
such a great question. There are so many groups of populations of heart patients that would benefit from cardiac rehab that it's not covered by insurance. Now, one group that is covered is heart failure. So if you have HCM and you do have heart failure, then you could maybe get it covered through that route. But if you have HCM and you don't have heart failure, it's going to be really hard. Some, like at our program, we are linked in with our prevention program, and so we have some access to trainers. So sometimes a personal trainer that is sort of tuned in to helping people gradually build up can serve in that role. But you're absolutely right. It's just it's criminal that insurance doesn't pay for cardiac rehab for a lot of these groups of people, including HCM.
So I have a dream. I don't have a lot of time for my dreams, but I have a dream. I would love if somebody who's newly diagnosed with HCM or long-term gets a family prescription for exercise. So they learn how to exercise as a family. If the parent has HCM, the child might be nervous for mom or dad. If the child has HCM and they're exercising, the parents may limit because they're nervous too. I think families should get together and learn what shortness of breath feels like when they're exercising, do it in a monitored situation so that they all feel safe and they can build some confidence. But I haven't figured out how to fund that one yet, so I'm working on it. And I look for partners. Hint, hint, nudge, nudge. Anybody want to play? Okay, so I posed an exercise question earlier. It may have gotten overlooked. Oh, Doug, I think it just answered it. Okay, and back to the myosin trials. Are they recommending? Okay, so the devil's in the details. There are 2 companies doing clinical trials in myosin inhibitors. Background therapy is permitted in all of the trials. There's a specific head-to-head with metoprolol and aficamten. So there's a couple different trials here. So the question is, are they recommending reducing beta blockers prior to starting these? John, you want to talk a little bit about the medication regimens on some of the trials you spoke of earlier?
Sure. So in the Maple trial, they're specifically looking at beta blockers versus metoprolol. Most of the trials, I think the metoprolol is left on for the trials. So, so that's so most of those are left on. A lot of them, there's kind of tight regulations and by the tight monitoring too, because they're 2 medicines that kind of work in concert. There's kind of tight monitoring by trying to keep people on a steady dose. So all for the trials. So it depends on the individual trials, but that's something that we're working out. I think looking in that was the specific question that's being answered in sorry, I think I said Maple trial. It was my trial's confused here.
Acacia.
Sorry.
The acacia? Oh,
No,
the trees. The trees. I can't. Cyto in their trees. I'm confused.
it was let me see. Getting late here.
You were right. You were right, John. You mean metoprolol versus aficamten? That's a case. Or sorry, that's Maple. Yep.
Yeah. So but I think that's the question. Is, do people need to be on both medications? I think that'll be so the data from that trial will be helpful. We've noticed in some people that we've kind of gotten to a maximum dose of the myosin inhibitors and needed extra metoprolol, or we've got other people too that were on a myosin inhibitor and then were on metoprolol for control of atrial fibrillation or something. They got an ablation for the atrial fibrillation, and that wasn't an issue anymore. And we took them off the metoprolol, and all of a sudden they had a significant obstruction again. We had to bump up the myosin inhibitor. So there's a kind of a complicated relationship between the 2 of them. So, but we'll get more questions. There are additional benefits being on metoprolol in addition to a myosin.
I've seen a great number of people lower their metoprolol after a long time on a myosin inhibitor, like months or years, and they slowly start to bring it down. So it'll be interesting to see how this plays out. I would encourage everybody to speak to their clinicians about their management and how they're feeling and the side effects of the drugs, and manage that with the disease in a transparent and maintain good communication the entire way through the process. That's what's going to make the difference here. I am going to say goodbye to our friends on YouTube, and I have one question here in the queue that we're going to answer. If you have a question that you want not on social media forever, I'm going to say goodbye, YouTube.